I'm a math PhD, and I got into this because I was reading uh, AI papers on the archive when I was in undergrad, and I was taking computer science, and I couldn't understand the things I was starting to read. I particularly got interested around the AlphaGo time, uh, just before AlphaGo, and reinforcement learning from David Silver. And they were doing things where they were playing Go. They weren't using convolutional neural networks yet, but they had some similar approach where they were taking local patches and trying to patch them together with neural networks and obtain some kind of spatial reasoning. And I thought that was very interesting. I've been a Go player for maybe 15 years, and it seemed like it was roughly state of the art at the time. So I tried to get into it, and the math was just a little much. So I started taking math classes, and I had during the pandemic, I had the Newton copycat year. I was 23. Newton was 23 during his pandemic, and so I just stayed inside and read a bunch of math textbooks and got a little ahead. And I got into grad school, and I got advisement from my PhD advisor is Dr. Andrei Shilnikov. And if you have studied dynamics, you'll know the name Shilnikov because the simplest chaotic attractor in 3D is the Shilnikov attractor. But that is not the same Shilnikov. That would be my advisor's dad. But they all come from this Russian school in Nizhny Novgorod. But at the time it was called Gorky. And that was the Andronov-Shilnikov school of qualitative theory of ordinary differential equations. But after the neuroscience revolution with Hodgkin-Huxley, the dynamicists took off with it and have developed it much, much more. And so we have a pretty complete theory of individual neurons now. And so I'm going to talk a little bit about what we know now, what the Russians have been doing with neurodynamics since the Princeton cabal kind of stole dynamics from them and ran off with it in their own direction. It's developed a lot since, and there are several active areas of research now that are pretty promising. So I'll talk about some of the structures that pop up, some of the main results in the theory, and hopefully give some of you who may not have a complete neuroscience background some idea of what to think about in this area. At any point, if you have any questions, just please interrupt me. You don't have to raise your hand or anything. Like I like being interrupted. If you've got something on your mind, just unmute and say whatever you want to. But you can find me on X at GeoGristle. I post pictures of stuff I'm doing computationally. Our lab work is, we've got like 20 computers in here. We're doing CUDA programming all the time. So like, it's a lot of simulation. But we have some wet lab interop. So some of the models we have are actually taken from an electrophysiologist that did some work with us a few years ago. So this video in the background is actually from him. This is a dendronotus sea slug, and the nice thing about it is that the neurons are pretty big inside, so you can get good quality recordings from them. And he left us with a lot of data before he left the lab. Before I go to the next slide, if you're a little squeamish, you can look away. It's kind of gruesome, but you've been warned, so I'll give you a second. All right, going on. So when I joined the lab here, Dr. Shilnikov made me go to his neurodynamics class. And on the very first day, he had us watch this video. What this is a decerebrated cat, or decerebrate cat. So this is from, I guess, Soviet physiologists doing tests. And so what they do is they cut at the bottom of the brainstem. I mean, if you cut into the brainstem, you're going to kill the animal. But it's alive. The head is separated from the rest of the nervous system, though, like permanently. And you see that they've got it suspended from two strings up top, and they've got it walking on a treadmill. And it's not receiving any kind of electrical input or anything. This is all just responsive, reflexive to the sensation of having the legs pulled back by the treadmill. And if you notice, the gait changes when the speed of the treadmill increases. So you might look at it, and immediately when you first see it, you're like, okay, well, the legs are just being pulled back and they're flopping back. Like if your legs fall asleep while you're running on a treadmill and you hold yourself up by handrails, your legs will kind of do this, but that's not what's going on here. You actually get complex gait patterns here, and it's confident stepping. It's not, it's not flopping and kind of rolling the ankles or anything. You have a walking pattern and then running. So what's going on? Well, there's no feedback to the brain. The brain can't coordinate what's going on. We think about the cerebellum in the back, the brain coordinating all these complex motor patterns, adjusting for orientation of the body, the orientation of limbs, but none of that can happen here. So what's going on is that all of the control is being done by the spine. And in humans, particularly, let me see if I can draw on top of this. And for the squeamish people, I'll move to the next slide pretty soon. But in humans, there's your brain. Looks like I can't draw on top. I can go back. That one's got a video too. There we go. In a human, most of your spine comprises white matter. So the difference between white and gray matter is the surface of your brain is predominantly gray matter. And what that is it's all densely packed neurons. And you're doing processing there. And so you need cell bodies because you need to have most of the tissue be computational. Whereas if you look at one of these pictures here, you can see this particular neuron here has an axon, and these can be basically arbitrarily long. Like they can be 1,000 times the size of the rest of the neuron. So the soma is where computation is actually done, and as well as the dendrites, these little spiny things that stick out. And so if you have a lot of these somas relative to the amount of axons, or the volume of neural mass taken up by axons, you can do computations. So in the spine, a lot of it's white matter. So the reason that the axon is so long is because it's carrying signals. It's not actually doing computations. So white matter carries signals, and it's sending signals from the brain predominantly down to the muscles or to other parts of the spine to do further computations. And in humans, you have three locuses of gray matter in the spine, and they look like bulbs. And in these bulbs, I'll go back, you are doing computations for locomotion predominantly and reflexes. So you have densely packed neurons all in there, and they're not projecting outwards for the most part. Some of them are, like you have some inputs and outputs, but most of them are doing computations internally. So the most common type of neural circuit in those bulbs is what we call central pattern generators. So these are collections of maybe 3 to, who knows, maybe a couple hundred neurons. And they generate rhythms. And you can control the current coming into them. You can send an electrical signal in. And if you have a small current going in, they'll generate a certain rhythm. And if you increase the current, maybe the rhythm will change. And there are thresholds at which these changes in rhythm occur. And so it turns out that actually this gait is controlled by feedback from sensory neurons in the muscles and on the paws of the cat that are giving feedback to a central pattern generator network. And depending on the intensity of the signals coming in, that central pattern generator network will produce different rhythms and then output controls back to the muscles. So all that's done in these bulbs. And it's pretty localized there. It's like, you can be pretty sure if you're going to trace up where the feedback control loop is going to in the spine, you just look in those bulbs. You don't have to really look anywhere else. You just trace through the white matter, go up to there, look in the dense forest of gray matter and pick out the neurons you're interested in. You can do some injection of current if you want to test if you've got the right neurons, and that's essentially what you do. So nothing about the brain is necessary for these complex gates. So to actually get to how we figured all that out, we need to talk about the structure of neurons. And a lot of what my lab has done in the past is study these central pattern generator networks. So a neuron is essentially a capacitor. And so it maintains a charge at the membrane. So it's a cell, so it's got this lipid membrane all around. That's what the outside is formed by. And then it's permeated, or it's littered with these channels and receptors. And I don't show any receptors here, but to get inputs, most neurons are chemically signaling. So they don't, you think about a neuron sends a signal to another neuron and it's electrical, you're just sending an impulse. But that's mostly not what's happening, actually. It is true, you're generating a nerve pulse, an action potential, and that's propagating down the axon. But once it gets to the axon, these vesicles in the end of the axon, like at what we call the synaptic terminals, the vesicles release neurotransmitters. You'll have glutamate, you'll have GABA, like calcium, a couple other things. And they'll propagate across. And you have things like cannabinoids, and those do some back signaling stuff, but for the most part, it's not that interesting when you get started studying this anyway. And those neurotransmitters are these red dots at the beginning of the animation. So they diffuse across a small gap. So you would imagine there's another neuron down here. And these terminals right here are very closely connected to, they're not touching, but they're very close to these dendrites on the next neuron. And these neurotransmitters, just these chemicals that exit by vesicles when the voltage increases in the presynaptic neuron, they diffuse across the gap between the synaptic terminals, and they bind to receptor sites on the dendrite, and that causes a cascading effect. It raises the voltage locally, typically. It can also decrease it. Glutamate, for example, increases the voltage, and GABA will decrease the voltage. And you can see here, you get stimuli. So here are a couple of excitatory stimuli. So that would be something like glutamate, increases the voltage. And if it's enough of an increase in the voltage, you have this cascade effect where the sodium, the voltage-gated sodium channels on the membrane will open, and then sodium will flow out very quickly. And once the voltage gets high enough, the voltage-gated potassium channels will open, and then potassium will flow out of the neuron. So sodium flows in, potassium floats out. That's why we call these like salty bananas. Helps you memorize which way it is. And it brings you back down beyond the baseline that you started at before you had any stimulus. And then the potassium is turned off after a time delay. And so then through the use of ATP, the sodium potassium pumps can work against the gradient of electrical potential to push the balance of sodium and potassium on the inside and outside back to equilibrium. So that's what an action potential is. And once you get an action potential locally, the membrane is structured in such a way with all these voltage-gated sodium channels, so that the depolarization is, you're at negative 70 millivolts typically. It goes up to just past zero, maybe up to 20 millivolts or 40 millivolts when you have an action potential, and then that ripples out, just like you throw a pebble in a lake or something, like it ripples out uniformly. It's very quick. It's so quick that usually people don't actually model it, but you can find some examples where people do geometric modeling of this, and you need PDEs to do this. So like it's much more computationally expensive, and it doesn't actually have that much of an effect on the dynamics. But it ripples out, and then it gets to an axon hillock, which is where the axon begins here, and then it starts propagating down very quickly. And these myelin sheaths, these are just insulators that help the current propagate. So that's all you really need to know about neurons to understand like the basic signaling mechanism. There are also electrically gated neurons, and they don't have neurotransmitters. There's just neurons that sit beside each other, and they'll have little channels that ions can flow through, like sodium, potassium, whatever. And basically, they're thresholding how fast the voltage of one neuron converges to the voltage of the other. And if you leave them alone for a little while, two electrically coupled neurons will converge to the same steady state voltage. But other than in ganglia, like in basal ganglia, and when you have like some kind of ganglion cyst in your hand or something, what that is you have a bunch of neurons coupled together electrically, and they all act as one in some sense. It helps increase the intensity of outputs, for example. But for the most part, it's chemical signaling. So in 1952, these two neuroscientists looked at the squid giant axon, and it runs in a squid, certain species of squids. It runs essentially from here down to here. So it's a very large neuron, and it's a little cell body here, a soma, and the axon goes up to here. And what it does is it controls the muscles in the mantle. Okay? So it's a motor neuron. So because it's so big, they can cut the squid open, and they can put electrodes in there and do measurements and inject currents and do experiments. So they developed, I want to say they developed patch clamp. That might have come later, but they developed schemes for actually measuring voltage current in neurons this way. And they found that these neurons act as capacitors. And so they just copied the capacitance equations, and they did some curve fitting to figure out some of the parameters in this model. Like, for example, if you go read their paper, what they end up doing curve fitting for is figuring out the power of these extra variables here. So this is like this is essentially the conductance of the potassium channels. This would be for sodium, and then you have some sodium and potassium channels that are always open. They're not voltage gated, and so that helps you restore equilibrium. And we call those leak channels, so that's what this term is accounting for. So that's always on, and it never changes. So what you do is you can set this this current to 0, and then you'll get. I'm a little afraid to write again, but here we'll try it. You just get this. So you have all these terms negative, and we call each one of these a different kind of current. So the first one would be potassium current. The second would be a sodium current. And the third is a leak current. And you can write this into a ordinary differential equation solver, a numerical solver, run cut of 4 if you want, Euler's method, whatever. And you can put in initial conditions and you can see what the neuron does and simulate it. And it's extremely accurate. Let's see. So they won a Nobel Prize for that paper. Not the curve fitting part. It was for the actual electrophysiology that they were doing that they won the Nobel Prize for. And nowadays we've kind of souped up this model a little bit. So we'll actually model how open or closed the population of, say, sodium channels is. And we just use a population model for that. So it's just a number, 0 to 1, that essentially records the proportion of the total number of sodium voltage-gated sodium channels that's open at any given time. And that will be a variable of the voltage for sodium. But you also have things like inactivation, which is time delayed. So you need some kind of dynamical variable. So each variable you introduce, each dynamical variable you introduce, causes you to have an increase in state space dimension. So some of these models are pretty high dimensional problems to study. This one would be, let's see, you have one variable here, Vm, the membrane voltage. And then you have another variable for each of these in M and H. So this is a 4-dimensional model. And we'll look at what those state spaces look like and kind of give an idea of how to do dynamical systems study in a second. But beyond this, we're not modeling right now anything about axon propagation. We're not modeling anything having to do with dendritic propagation. It turns out actually that dendrites do a lot of computation. Intrinsically, they don't need to interface with with somas or other neurons or anything. They intrinsically have their own computations. And in some of the cells, like the one from yesterday, the pyramidal cells where dendrites are really dense, there's a lot of them. The majority of the computation is done by the dendrites. So it's fairly complex. I know there are a couple of people who study it, but it's not as well mapped out as just general neuron action potential propagation dynamics is. But you can study a couple of things. You can study PDEs for how the action potential propagates down an axon. Essentially, that turns out to be the heat equation. That's this. It's the cable theory equation, or the cable equation, I think they call it. And you can study if you have a bunch of white matter tracks running side by side, say in the spine. They're not insulated from one another completely, so you get some kind of dynamics from simultaneous propagation across multiple of them at the same time, so you can study that. And you can also study relatively innocuous things in the axon itself, like solitons. So axons do not discriminate which way they send signals. You can send a pulse right here, and it will backpropagate up through the neuron. That does happen sometimes in biological neurons endogenously, but it's not responsible for anything that we know is like a majority of any process that goes on in the brain. We kind of ignore that. And this picture here is the state space for one of these neurons. So here on the vertical axis, you have voltage. That's one variable, and we can model that as the real line. So -60 would be maybe like an equilibrium voltage or something. And then when you have a spike, maybe it jumps up to 20. So you can watch these blue trajectories. So this is a time series, actually, and it'll move around in this three-dimensional space. You might have like a calcium variable that tracks the membrane calcium concentration inside versus outside. And you could have some other stuff like in this model, actually, the X variable on this axis is accounting. It's a mixture of several ion channels, like an abstract ion channel. And it's a mix between slower and faster ion channels dynamics. So you'll move around in these 3D spaces, or higher dimension if you want. For this model, it'd be 4 dimensions. This one is actually 5-dimensional, but I'm hiding 2 of the variables because they're really fast and they're not that important for the overall dynamics. So you're moving around as time goes on. Your calcium is increasing, decreasing, so you'll move left and right. Your whatever ions are responsible for the X abstract ion channel, they're moving this way. And then voltage is going up and down. So every time you spike, you're going to jump up on the top of this thing we call a fish in the lab. I didn't come up with that name. Shilnikov likes to give things funny names so that we can refer to them easily. But they'll jump up on this fish, and then maybe you'll have a few spikes, and then the trajectory will hop off of the fish, and you'll go onto the dune, which is, this where you're not spiking at all. You're kind of oscillating around some kind of equilibrium, and you'll have quiescence during that period. You won't be sending signals to other neurons, even though you might be oscillating in voltage a little bit, calcium and all these other things. So these are the pictures. These are the phase-space portraits that are sort of central in dynamical systems study. So we'll look a lot at those. One kind of interesting thing about dynamics is there is actually a distinction that can be made between biological systems and mechanical systems. So, we had the Industrial Revolution. A lot of dynamicists, control theorists were going into making machines. And it turns out that, machines made out of linkages in the plane, motorized machines, things like we'll see like the cart pull, reinforcement learning, gym environment, pendula, all these things are mechanical systems, and they tend to exhibit what we call supercritical bifurcations. So what a bifurcation is if you look back at the previous picture, we have these structures in the state space. The fish, the dune, and then you've got an equilibrium here, and maybe you have some equilibria elsewhere, like maybe one up here. You can't see them unless you actually start your state at those configurations, exactly at those points. And they, if you change the parameters of the model, like let's say maybe you change this maximal conductance for the sodium channels, GNA. You'll change the topology of the state space. You'll move around the equilibria and any kind of periodic orbits. So those things will move around, they'll vanish, new ones will be created, they'll morph into other types of structures. So every time you have a topological change, not a geometric change in the state space, we call that a bifurcation. So if everything just was shifted to the left a little bit, that would not be a bifurcation. But if you create a new equilibrium, or you delete one, or you create a periodic orbit, or create any number of a bunch of other structures, those actions are called bifurcations. And so changing parameters does that. So supercritical bifurcations are when you let's say you have a stable equilibrium. So what that means is if I drop a ball into, like, I can take a dome and I can drop a ball in there, and wherever I drop it, it's probably going to just roll down to the middle due to friction. It's going to lose energy and converge down to the bottom of the bowl. So there's an equilibrium there in the state space. I can imagine what my state space would be. I've got a ball, it's got a position, it's got a momentum, and it's moving around in this 6-dimensional space because you've got 3. You could actually do it in 4 dimensions because you're moving around in 2 coordinates on the bowl, on the surface of the bowl. So you got like an x and y coordinate, and for each of those, you've also got a momentum, or a velocity if you want. So you've got 4 dimensions in your state space. You can imagine a 4-dimensional space, and as the ball rolls around in the bowl, the trajectory will move around, and it will converge to an equilibrium at position 0 with momentum 0 because of friction. So that we call those stable equilibria if things nearby them converge to them. And you can imagine a pendulum. So like, let me draw it actually. Two fingers. A pendulum. So you're living in the plane and you've got a stick and then you've got a massive ball on it. By that I mean it has weight to it and it can rotate. So a pendulum has, let's suppose there's some friction in the hinge where pivot occurs. It has a stable equilibrium here when the mass is hanging down. So, if I perturb it a little bit, this is going to swing left. And then it's going to swing back to the right, and it'll go a little bit less far to the right that time. And then it'll go back to the left. And the oscillations will decrease in magnitude, and it will converge to this steady state position. So that's a stable equilibrium. On the other hand, you can imagine that you perfectly balance the weight straight up, and the pendulum will not ever move if you do that, assuming there's no wind or the joint isn't jittering or anything due to like Brownian motion or an earthquake or whatever. So if you do that, but you perturb a little bit, you just barely nudge the pendulum, it will obviously fall back down. So you just nudge it a little bit and it will just fall and then come back up here, and then it'll go back up this way and then back down, and it'll eventually converge back to the stable equilibrium. So we have stable and unstable equilibrium. So supercritical bifurcations are when you start with stable equilibrium and you change your parameters, you change your model in some way, and at some point that equilibrium becomes unstable, and out from it grows a limit cycle, or a periodic orbit is what we typically call that. So you get some kind of oscillatory behavior. So if you're near the equilibrium, I might be nearby it, and then I change my parameters, and I keep changing them. And then I get out to here, and I start swirling away from the unstable equilibrium. And I'll converge to the stable limit cycle. And I'll get asymptotically close to it as time goes on. So mechanical systems have this type of behavior to them. Okay. So this would be an example. The bifurcations occurring are when latches let go through some kind of electrical impulse, and the parts in the machine will stop being stable. They'll stop holding still against some kind of barrier that's holding them. There's a lot of force being applied in the system holding it still, but when a latch is let go, like this one here or something, you get a limit cycle, and so some of the other parts will start moving, and they'll oscillate if they don't have any more barriers to run into. They'll just go around in circles. But they run into barriers, and then you get another super critical bifurcation in reverse. So you're killing off the stable limit cycle, and you kill it by running it into an equilibrium in the state space that's unstable, and then the equilibrium becomes stable. Now, vice versa. If you start with an unstable equilibrium, so we're going to go this way this time. You start with an unstable equilibrium. If you were near it, you would go away from it, and you would just go off forever, basically. But you can have other structures elsewhere that push you back or nearby and keep you from running off to infinity. But with a subcritical bifurcation, the unstable limit cycle will switch stability to stable, and then out from that will grow an unstable limit cycle. So if you were to start here, you would circle around and spiral in on the stable equilibrium. So biological systems tend to have this, and the reason for that is because you want to have the ability to switch modes of activity very easily, and you want to have a lot of optionality in your state space for where trajectories end up. And part of this is evolutionary. Like you don't want to have a scheme for bifurcations that requires you to carefully design the state space. It should evolve naturally, and there should just be a lot of structures in the state space that can be opted between, and then evolution can prune out the bad structures. So you're always kind of teetering between unstable structures in biology. So you have all this activity all the time. It would be more efficient if I just wired up some kind of electrical circuit that did whatever computations I want to do, and there's one wire per type of signal, and it just sends a signal when an event happens. But that's not what's going on. Everything's sending signals all the time. They're just changing magnitude. And so you want to be able to switch modes overall. Mode switches should propagate across networks very quickly, this sort of thing. So in general, you tend to see biological systems having subcritical bifurcations rather than supercritical. So in the 50s and 60s, the control theorists and dynamicists figured out pretty well how to, let me pause this because the audio. There we go. So the dynamicists figured out how to take unstable equilibria and use them in state spaces for mechanical designs in combination with computerized control. So you have all these sensors, like so let's say on an airplane, or you could even take the pendulum example. You put the pendulum standing straight up, so if it is perturbed a little bit or you don't get it exactly perfect when you stand it up and balance it, it'll fall over. But it's easy because nearby an equilibrium, things move slowly. It's easy to have a computer control it and keep it upright, even if you don't have much torque in your actuator. So a computer can just monitor the state and then control near unstable equilibria. And then this is nice because it kind of gets the benefit that biology has, where it's easy to switch modes really fast. So if you've ever seen the Top Gun movie, the more recent one, they like half the plot is about these fifth generation fighter jets. And so the point of them is that they fly in a steady, unstable equilibrium. Like their heading is unstable. So if there weren't a computer on board, the plane would just wobble really quickly and then flop, and it would basically belly flop through the air, and it wouldn't maintain its heading. The reason for that is because it's advantageous to be able to kill off the artificial control of, like the computerized control of the maintenance of the unstable heading. And when you push your joystick forward, the plane doesn't have to fight drag to turn over a long circumference for like turn radius. It can just turn, and then you have aerodynamics pushing you down, and so you can just turn and go down instantly. So you can see kind of an example of one of these here. You have good heading control even when you're not, even when you're stalling, essentially. So they're designed to take advantage of that. The cart pull example from reinforcement learning is similar. You're just doing computerized control to keep you in an unstable configuration. The idea is that you have a cart, you have a pole you want to keep standing upright, and you keep it that way by driving the cart left and right, and you've got to do it delicately enough that you don't run into the walls. And that's the only actuator you have is the wheels. You can study things like the controllability of unstable systems like this. using Lyapunov functions, which are analogs of energy functions. So you can measure energy levels in the state and say, I don't want to let the energy get too far away from where I want to be, because then I have to expend that much energy at least to get it back. So you can put like bounds on how far away you're going to tolerate the state to get from where you want to be due to energy or the Lyapunov analog of that. And let's see. So here's an example from neuroscience. I think this is a chicken locomotor neural network, and we're looking at, so this is a pacemaker network. So 2R is coordinating the action of two other neurons. And when it fires, the other two go off. So it's inhibiting the other two. And that happens through network effects. It's not a direct inhibitory coupling or anything. But when it fires, the other two go off, and then it will lose its firing will lose its power, and then the other two will be able to turn on again. And then they turn on, and then that turns 2R back off. And so you get this oscillatory pattern where 2L and 3R are firing, and then they alternate with 2R repeatedly. And so you can have them all project out to legs or something, and then maybe 1R and 3R project to different parts of the right leg, or maybe the left leg, who knows, or different legs even. But they will control an alternating gait. So that lets you, this is just like a canonical example of a central pattern generator. But if you inject the TTX neurotoxin, you can see that you're killing off some of the control mechanism in the neural circuit, and it causes everything to just fire erratically. So you're losing some of the communicability between the neurons in the network, and then everything just falls apart. You just get erratic firing. There's no predictability, nothing. You can't control it anymore. So this is like, you don't have this nice bursting pattern from here anymore, where you're on, off. It's just a bunch of spikes and then some erratic fake quiescence here where you're still spiking a little bit. So this actually is a model for neural chaos that kind of spawned off a lot of the neurodynamics research after it. So how do you actually study these state spaces? Well, the main thing you look at first is the equilibrium. Let's go back to the equations. I have these four variables. I've got the membrane voltage, Vm. I've got N, M, and H. And I've got these equations that I've got a four-dimensional system of autonomous ordinary differential equations. And so I can just set the derivatives to 0. And if the derivatives are 0, then the variables aren't going to move. So I'm going to be in an equilibrium. So that's what these dots are here in the state space. So in this example, I've got two variables, one on the x-axis, one on the y-axis, and you can see these equilibria here where the state isn't moving. So you can see the trajectories too, so you can tell like the vector field that controls the flow, which is coming from the differential equations, that is going to be 0 here and pretty close to 0 here, so you're going to move slowly. So you'll go quickly through here, and then you'll slow down and asymptotically approach this stable equilibrium. And here, this one's unstable, so if you start super nearby, you'll move slowly, but you'll go away from it and then speed up. So you just set the derivatives to 0 and solve the equations. Now, if you don't set all of the equations equal to 0 simultaneously, you get what are called nullclines. So if I just set like the x derivative dx dt equal to 0 in this, and then solve for the curve that would give me whatever the equations for this model are, I'll get, I'll get this dotted line, and this one, and this one. And so if x dx dt is 0, I'm not moving in x, so I should only be able to move in y, which means I'll go up or down. So nullclines tell you when are you going to only be moving vertically. So every time you hit this curve, you'll be moving vertically. And you can use like Fermat's theorem and say, okay, well, X is going to be at a minimum or maximum when I hit these null lines. So the other kind of important feature to look at is like they also separate the null lines separate which directions you're going to move. So if you're above this curve, you'll be moving to the right. So you're always going to the right in this region. If you're out here, you're in the same region, so you're going to still be moving to the right. And if you're over here, you're going to be moving to the left because you're on the other side of the curve. So these split the plane into separate regions. And then up above this one, you'll also be moving to the left. And you have the same thing for dy dt equals 0. So you're moving down on this side, up on this side in this region, and then down in this region. So these null lines make it really easy to study the dynamics because you can basically just throw away the trajectories and then redraw them and get pretty close to what the original was. And you can also see the equilibrium in them from where the null lines cross each other. It's another example. Some of the equilibria you might spiral out from, and some you might just go out in a linear fashion or in towards, doesn't matter. And in higher dimension, I have some software that you can try if you want, and there's a link to it. But you can compute null lines in higher dimension, and they turn out to be hypercurves there, or hypersurfaces there. So in 3D, your null lines will be surfaces, and this is the dz dt equals 0 null line in black. In blue, I've shown a trajectory, and then in red, you're actually showing a trajectory live, or a bunch of trajectories. And when you're on the black surface, you will not be moving up or down. And when you're outside, you're going to move down, and when you're inside, you're going to move up. So makes it very clear what the structure of the state space is, or at least it gives you a starting. And in neurodynamics, you have a particular null line structure that essentially always appears, and it's a cubic null line with a linear null line passing through it, so intersecting it. So let's see, this is you're going horizontally when you cross the cubic one, so that would be the dy dt null line. And this is the dx dt, so you're going only up or down when you cross it. So this structure lets you go around in an oscillatory fashion, so you can simulate things like bursting and spiking. You can imagine the y dimension is like voltage for a neuron or something, and so this would be at the top of a spike up here, and then this would be down when you're quiet. And this model fits Unigumo. They literally just took two equations, dx dt equals a line, and then dy dt is a cubic, and that's all they did. And if you're interested in studying things like null lines and other structures in the state spaces, this book is really nice. I don't see it recommended much, but it's essentially the most comprehensive. Okay, so now we've seen the null line structure that appears in neuron models typically. I should say, like, the Hodgkin-Huxley model, the 4-dimensional one, that one also has that cubic null line, linear null line structure. So it appears in physically inspired models. It's not just how can we simulate things that look like neurons. It's actually, if you try to model the physics of a neuron, you get that structure. So we can then study, well, what if you have a neuron that does bursting? So sometimes it's quiet, and it'll oscillate between quiet and active. But when it's active, it's also going to spike multiple times. So that's a burst. And the spiking should be some oscillatory pattern too. So you need two coupled but separate oscillatory patterns in your state space. So Ajikovic, I think he's Russian. He comes to the lab sometimes, but I haven't gotten to meet him. I was out the time he came last. But he published this really nice book that kind of introduces neurobiology for dynamicists. And what I know him best for is his classification of bursting regimes topologically. So he has pinned down these 4 ways that a bifurcation can occur such that you start spiking in a burst, and how a bifurcation can occur such that you stop spiking in a burst and go back to being quiescent. So you have fold bifurcation can start a burst, a SNC or saddle node on an invariant circle bifurcation can start a burst, and then you can have supercritical and subcritical Andronov hops where you turn an equilibrium into a limit cycle, and then you can have the same things or similar to them for ending a burst. And the idea is you take the plane and you construct the Fitzhugh-Nagumo model where you've got the cubic nullcline, or maybe even a quintic nullcline, and a linear nullcline that goes through it. So you get this hysteresis loop. And then you add another variable, and then you couple it to the other two. And this allows you to construct some kind of bifurcation structure because you'll have oscillations in that third variable. And you can pretend that the parameter for that third variable, and maybe the second too, this one, is controlled by this variable going this way. So that variable, even though it's a state space variable, acts like a parameter for the other subsystem. So we call this plane where the spiking occurs the fast subsystem, and then we call this one where the bursting occurs a slow subsystem. So you can kind of see the recipe book, or the recipe for doing this. And you'll find if you study real neurons, they all tend to match one of these schemes in the classification. You model the biology physically, you look at the state space, you can figure out what the bifurcation structure is, and it'll match one of these. Some are more common than others. Like I think for the ones I study, typically fold circle is most common. So this was a pretty big result in neurodynamics. Like we got a complete classification for bursters. And bursting is essentially the most complex type of behavior that a neuron can do, really, in the soma anyway. So how do we go on from here? Like what else is there left to do if we've classified every way a neuron can burst? Well, it turns out we can actually get a universal structure that describes all neurons. So I want to talk about that. That'll be the second point of this talk. And then we'll have a third at the end, but that one will be a little different. So we have attractors in these state spaces. So you have some differential equations. Maybe they describe a physical system that loses energy over time. Like you have friction in the pendulum model. In thermodynamic systems, you might not have a closed system. You might have loss of heat over time. And so volumes in the state spaces will get contracted over time. They'll shrink. And so if you're shrinking the volume, if you take a volume and you evolve it in time and watch all the points in it move around in the state space, and it tends to shrink in volume, then it's going to have to either run off to infinity, right? Like just diverge. You can have the volume shrink still doing that. Imagine you just take a cube and you shrink it as you send it off in some direction. It can go off forever. But if the states are confined to some region in the state space, as we want for these biological models, because the calcium only stays in a certain range for a neuron, you're going to converge to some attractor. You're living in a compact space, and you've got a contraction, so you're going to converge to some set. So you have attractors. The states in the system will converge to these attractors if you lose energy. If you don't lose energy, if you're doing Hamiltonian dynamics, it doesn't happen that way, but otherwise, yes. And these attractors are topological subspaces of the state space. So we want some way to study those. Should we think of them as simplicial complexes? Should we think of them as manifolds? Should we think of them as cellular complexes? Are they something else? Who knows? So it turns out that for the most part, we have something in between cellular complexes and manifolds that is a very good description of dynamical attractors. And I'd actually say all of the continuous dynamical systems that you see are described by, like in the literature, typically are described, all the famous ones, they're described by these templates. So it's really useful to figure out how they work. Well, what it is you just look at where the trajectories go, and they'll fill out this attractor, and you can really see it if you just draw the trajectory. That's what these curves are. And then the attractor turns out to be where these trajectories converge to. And this is some subset of the attractor, but the blue is a cellular complex. And it acts like a manifold. It acts like a surface. But the problem is, this looks like a surface too, but you have these branches that are themselves surfaces. So if I took this little ear off, I would have a surface by itself. It would be like a cylinder. But you join it to another ear in this way, and you lay them down flat right here, and you get singularities. So this isn't a manifold, because this part right here looks like if I cut out this region, it would look like a Y shape. That's not a manifold. So what's the trouble? Well, the tangent spaces where you join pieces like strips from the surfaces, you join them, they're tangent to each other when you join them. You're like laying them down and putting a piece of tape on top and pressing it down. So what we get is called a branched two manifold. So there are joining loci that adjoin multiple branches, and the tangent spaces of them overlap. They are the surfaces that are conjoined are all tangent to each other where they're joined. But otherwise, you have a surface everywhere else. So the joining locus for this template is right here. And like the joining locus for this template is right here. And then you have things like splitting here. And then pieces of it can fold over. And they can be arbitrarily complicated. Like you can see down here. This one, I believe, comes from some combinatorial problem, not a dynamical system. But you actually see this one in Shilnikov attractors. And this is Robert Grist's universal template. But the talk isn't necessarily about templates, so I won't go into that. But this was like kind of a bombshell result in template theory. It said, you guys are all studying the wrong thing. You're studying the knots that live in the templates, but you shouldn't, because there's you're trying to classify attractors by what knots their templates have in them. And it turns out that there's actually a pretty proliferated set of templates that have all knot and link types in them. And so there's no hope to classify attractors by the knots that live in them. So instead, you should study the templates directly. So it's actually more important to look at these templates. And you can describe them algebraically by writing them in what we call standard form. And standard form just says, okay, break it into 4 pieces. So you have parts that just flow laminarly. So if you start here, you'll just flow around, and you'll come back up to here, and you'll go onto one of these branches, depending on where you come to at the splitting line. And you go down one of these branches, and you might have orientation reversals, and it might be clockwise or counterclockwise that it's twisted. And then things can cross over each other. And then when they insert at the joining locus, certain branches will be in front of others, and certain ones will be in back. So you have this like filing cabinet structure. And it's a question of which one's in which order, right? So you need to describe that if you want to talk about a template. So you give 3 things. You say, what are the twists? What are the crossings? And then what are the insertions into the joining locus? And that's enough to describe what a template is. Okay. And the other thing Robert Grist did was, when he developed this universal template, he gave a really powerful topological tool for manipulating templates and showing that multiple are equal to each other. So they're isotopy invariant, so you can move them around in like 3D space. And as long as you do that and you don't like tear them or pass them branches through each other, it's the same template, but if you do like a diffeomorphism or a homeomorphism, it's you actually, or a homotopy or anything, you lose the equivalence of the template, so you actually change what template it is. But inflations are a nice trick that let you show equivalence without having to do isotopies only. It's like an algebraic equivalence. So what if your attractor isn't two-dimensional? Well, we have another tool for that. You just take this idea that the template was a bunch of two cells conjoined, and you say, okay, well, if my attractor is four-dimensional, I just take a bunch of four cells and conjoin them. And you get these things called templexes, and there's a computational algorithm for getting them. And these work for a broad class of problems. They came from this Argentine research group, Cimarella, and I don't think anyone else is using them. But if you're interested in that sort of thing, chaos topology, like this would be a cool avenue to go down. The trouble is that all the literature is in Spanish, so you have to translate it if you don't speak Spanish. But fortunately, most of the neural templates are 2D, or the attractors are 2D, so we can just use templates. So how do you actually get them? Like if I have a neuron, what do I do to get a template? Well, I can numerically simulate a bunch of trajectories. So this is in a California sea slug. It's one of the neurons that controls the motor reflexes. And I'm starting a bunch of trajectories on a line. Here's an equilibrium. I draw a line out, and I start a bunch of trajectories on that line. And I ask, okay, well, if I start at a certain calcium value on that line, when I evolve the trajectory, it's going to spike a few times and then come back and become quiescent, and then it'll flow down here, decrease in calcium, and it'll hit the line again. When it comes back to the line, you record where you started and where you ended, and you can plot that in a graph. And you just do this for all of the points on that line, and I could fill it in more. I would get like a curve, but you'll get a graph. And you can evolve this graph by just iterating the function starting at some point. So you start here, and then you'll take an iterate of the function, and it'll send you up to here and then over to here. And then you'll iterate it again at this point, and you'll go up and then to the right. And then you'll go down and to the left, and then up and then to the right. And so you can follow the trajectories just from this map and study the dynamics, and it's equivalent, actually, to studying the ODE. So it's much cheaper when you get this map. It's a little bit numerically inaccurate, but it's serviceable. And you can study the structure of this map to figure out the interesting dynamical features instead of having to study the topology of the state space. And it will give you clues about the topology too, so that's nice. So you do this, you figure out kind of what are the pieces of this return map, this graph. And you should get a template for this system that has a branch for every monotone piece of this graph. Okay, so wherever it's decreasing, you should get a branch for that. Wherever it's increasing, you should get another branch from that. So you should have 1, 2, 3, 4, 5, 6, 7, and so on branches. And it turns out it's actually infinitely many branches, so you'll have a problem in a minute. But you use this information to help, because I'm living in 5 dimensions and I can only really see 2 or 3. So I need to use computational tools to help me figure out the hidden structure here. That's it. Oh. So yeah, you draw out the topology, you try to figure out what it looks like in 5 dimensions. And then you piece together the parts of the template, like the reinsertion order into the joining locus and all this nice stuff. And it's very hard to do because you have to like unwrap the attractor, like you have to fold it and figure out the way the branches cross over each other and all this stuff. So it's helpful to use paper models. Once you get an idea of what the branches are from the return map, you can cut out strips of paper and then use that to do ambient isotopies. So this is really nice. So then you get a pretty complete picture of what the attractor should look like, all the topological features in it. and you get a template. So the trouble is, this only describes 4 spikes in a burst. So if I hit this branch, I don't spike. I just do a subthreshold oscillation without spiking. If I hit this branch, I do 1 spike. If I hit this branch, I do 2 spikes, either of these branches. If I hit either of these, I hit 3, and so on. But I can have parameters change so that I can have any amount of spikes. So this template doesn't describe all that. I could just keep drawing the template to the right, but then it wouldn't be compact, and I couldn't call it a template anymore. Also, some other authors in '99 guessed that a similar structure was responsible for neural bursting. And they didn't get the whole picture here. They didn't study anything that was going on away from what we call the fish, where the spiking actually happens. So it was necessary to really figure out what the total topology was. So the trick is you can take this template here and you can do a template inflation. Thank you, Robert Grist. And you get this compact template that describes any number of spikes. And it tells you what the topology is of the attractor. So this gives you can label all your branches, and you get a semantic alphabet for events that occur in the dynamics. So A is like a subthreshold oscillation. You start a burst at B, you do a few bursts on D, and then you end your burst, or you do a few spikes, and then you end your burst with a spike on C, and then you can come back with an orientation reversal or not. And then you just start over again. You have a oscillation on the dune. So that's a subthreshold. And then you can look at voltage traces, and it actually turns out that each of these symbols corresponds to a part of the voltage trace. So you can recover your symbolic sequences and figure out what your 5-dimensional continuous dynamics is just from these voltage traces or from these symbol sequences. And it turns out you can also put an order on the symbolic sequences. So they're ordered from least to greatest. So you can say which ones are closer to each other than others, or you can say how far they are and all this nice stuff. And you get a scheme for talking about the order of symbolic sequences. And the big result here is neuroscientists like to take voltage traces and study them by chopping them into fixed time bins and asking how many spikes did you have in that bin. You'll take little slices here, and maybe you didn't spike in a certain bin, and so that would be 0 spikes. And then this one, maybe you have 3 spikes in it, and so you call that 3. And they will then study the complexity of the numerical sequences that come from this time binning thing. And they've done this since they've been able to do EEG, or since they've been able to do local field potential measurements in individual neurons. But this shows that they're not actually using all the information available. There is one more bit of information in each burst, and that is the orientation reversals that happen when you come back on E or F. So you can augment the spike count binning by adding a sign. So you say, if you have an orientation reversal, you put a negative and you keep a positive if you don't have an orientation reversal in that burst. And then this lets you actually talk about voltage traces that are close to each other versus far away. And you can study how far you are from chaos, for example. Okay. So once you get those return maps, you can use needing theory. So if you can use finite limited points in the map to study the entirety of the map and get global information. You just study the critical points. And you iterate them forward, and you track which monotone pieces they land on each time. And you write out this symbolic sequence. So you would say like lap 1, lap 2, and then this itinerary would go lap 2, lap 1, lap 2, lap 1, and then it repeats. And you can recover things like topological entropy and detect if there's chaos anywhere in the map just using a few points from the map. And here's some software for actually doing this. I think this is the only implementation that's available. So using needing theory, we can take our 1D maps and we can take voltage traces, compute their sign spike count sequences, and then recover the needing invariance for the 1D maps. And this is going to be useful because we'll be able to compute the topological entropy of the ordinary differential equation system. So as far as I know, this is the first time somebody's computed topological entropy for any ODE system, but it's extremely cheap. I have a paper, or excuse me, I had a presentation in Denver, and that's what this QR code takes you to, but it shows you how you can actually make this cheap. It's computationally intractable if you just do what I said in this talk, but there's a really nice trick using geometric singular perturbation theory that lets you make this ultra fast. It's faster than any other method for computing complexity, like even faster than maximum Lyapunov exponents. And it gives you global information, whereas other things only give you local information. So much, much better. And the state of the art symbolic dynamics, I guess, or in complexity for ODEs. And you can plot things, like you can sweep over parameter spaces and say it's complex behavior in this region and simple behavior in this other part. So it's very fast, so you can do this quickly. All right. I may have to skip a few slides since the time's low, but let's see. Yeah, okay. Well, I think I got 2 or 3 slides that are salient here. So the Shilnikov school in Russia put forward this conjecture that said every attractor is either pseudo-hyperbolic or quasi-stochastic. So if you're pseudo-hyperbolic, then you are robust. You're robustly chaotic with respect to perturbations. So if you change the parameters a little bit, you're going to stay chaotic. If you're quasi-stochastic, on the other hand, you will become stable with a small perturbation. So you break your chaos very easily. Most things are quasi-stochastic, but things like climate models, for example, are pseudo-hyperbolic. So the canonical example of a quasi-stochastic attractor is the Shilnikov homoclinic attractor. And all you need is an equilibrium. You need a trajectory that starts and ends at the equilibrium, and it spirals out, and then it comes back in on linearly. So you can do this in 3D. And if the speed that you come back is faster than the speed that you go out, you will have countably infinitely many unstable periodic orbits and chaos. So that was what Shilnikov became famous for. He proved this using what was called the Shilnikov map. So another application of the 1D maps. And this is ubiquitous. Like anytime you have oscillations of any kind in any dynamical system, you're going to get these. You even have them in like economic models, for example. So quasi-attractors appear in neuronal systems with a little more structure. So you'll have that Shilnikov homoclinic, but you also have some other structure where you'll have a Hopf bifurcation, subcritical, so that you grow oscillations out from your equilibrium. And this studying this was really necessary to figure out what the template was. It wasn't possible to go further than Gilmore did in '99 without actually figuring out what the structure of this Shilnikov-Hopf bifurcation was. Now, on the other hand, pseudo-hyperbolic dynamical systems come in 2 forms. They can be continuous, like ODEs, or they can be discrete maps. And there's only one known example that of an ODE system that has, let's see, I should say what is called wild chaos, which is an advanced form of pseudo-hyperbolicity. And it's this 4-dimensional Lorentz system. And they're using needing theory to figure out that it's wild chaos here. And Bonatti and Diaz show that you can construct wild chaos very easily in discrete maps by using what's called blenders. So that's similar to smell horseshoes, but a little bit augmented. So for studying any of these systems, quasi-stochastic or pseudo-hyperbolic, you want to use Lyapunov exponents, and they're just numbers, one per dimension. And they tell you what the rate of expansion and contraction in each direction is. And so if you have any expansion at all, you're going to have chaos. So if you have a positive Lyapunov exponent, you have chaos. You can also use these numbers, these Lyapunov exponents, to compute the dimension of the attractor with this formula. You look at how many of them from greatest to least in that order are required to get a negative number by summing them, and then you sum the rest of them and divide by one of the exponents, and then you take the sum of those two numbers and it gives you the attractor dimension, and you'll get some fraction. So it'll be, for Lorentz, it's like 2.1 or something like that. It's fractal dimension. And to go further than the Lyapunov exponents, we, in 2007, not me, but Janelli gave an algorithm to actually figure out what direction your expansion and contractions are in. These are called covariant Lyapunov vectors. And these live in the ambient tangent bundle over the restricted to the attractor. They're frames, and they're global frames in the attractor, or at least on a covering space of the attractor. And so they're covariant with respect to the dynamics, so you can actually compute them very cheaply once you get the starting one, and you can evolve them across the whole attractor and figure out what they're going to be everywhere. And let's say if you started at a point and you step in the direction of the red arrow, you're going to maximally change the future trajectory. So if you want to start somewhere and you want to get somewhere else other than where you're going to go if you don't do anything, step in the direction of the red arrow. And if you're starting somewhere and you want to change where you are right now, but you want to end up in the same place, step in the direction of the green arrow, the contracting direction. So it turns out, using these covariant Lyapunov vectors, you actually have a criterion that lets you figure out if an attractor is pseudo-hyperbolic or quasi-stochastic. I think this was 2020 that we figured out that you could do this. And it's so cheap. Like before, you had to use cone conditions in the tangent, or excuse me, in the ambient tangent bundles, and it was extraordinarily difficult. Only theoreticians could do it, but now anybody with a computer can do it. So, for example, Lorentz, you ask does the contracting subspace and the non-contracting subspace, do they ever collide with each other? If so, then you are quasi-stochastic, and you see that the minimum angle between those two subspaces hits 0 right here. So that is a quasi-stochastic system. But here, that never happens, so you have separation between the two subspaces, and you are pseudo-hyperbolic. So if you perturb the one on the right, you shouldn't have stability, and you shouldn't lose chaos. But if you perturb the one on the left, you'll lose chaos. You change your parameters a little. So here's some more examples. Rossler, Lorentz, Shimizu Morioka, another Shimizu Morioka here. You can see where the vectors lay down flat, so they collide subspaces. And finally, I want to pose an open problem. Can neuronal attractors be pseudo-hyperbolic? I've been looking for a bit, but I have ideas of mechanisms to generate pseudo-hyperbolicity in neuron models. But even though I get traces of evidence that I've created pseudo-hyperbolicity, I go and actually check with the covariant Lyapunov vectors, and that I don't find that it's pseudo-hyperbolic. I was talking to the guys that developed this algorithm last year, and they said that they thought I should be able to even get wild chaos because I have these heterodimensional cycles in my template, and I should be able to carefully pick out parameters where you get heterodimensional cycles, and then the theory says if you perturb a little bit from there, you're going to get wild chaos. But I never see it, and it should be open sets in the parameter space. So it's hiding, and it would be extremely useful to have a constructed model of pseudo-hyperbolic neuronal chaos, because every time you perturb a chaotic neuron that we know of, chaos dies. You can simulate it, you can fake it with noise coming into the neuron, noisy environmental conditions, but you can't robustly maintain it intrinsically. So I want to pose that as an open problem if anybody's interested in this area. And I suppose I'll take questions. Thank you, guys.
Thanks, Carter, for the talk. Sorry I had to rush you. So we have a question in the chat. Can you please add here a link to the paper on fast complexity estimation?
Yes. Yeah, it's a blog post. I'll send it.
Okay, great. I was actually kind of wondering about you said that there was a conjecture of Shilnikov, and I was kind of wondering what you know about progress on that conjecture.
The P or Q conjecture?
Yeah.
So that one is by Shilnikov's student, Sergei Gonchenko. And that was made in the paper where they figured out that you could use covariant Lyapunov vectors to detect pseudohyperbolicity. And yeah, so if let me grab this link real quick, just so I have it in my clipboard. So if I go back, I put a little note in there talking about what it would look like to resolve this conjecture. So if you wanted to resolve the conjecture to the negative, you would need to give an example of an attractor that is not pseudohyperbolic, but which is still robust to perturbations. So you should find in the parameter space of, in the space of dynamical systems, you should find an attractor that is not pseudohyperbolic, but it lives in an open set in the space of dynamical systems where all those systems in that open set are chaotic. And that would resolve the conjecture to the negative. I don't know you would have to give like some kind of logical argument for the resolution to the positive. Everybody just assumes it's true. Does that answer it?
Yeah, that answers my question. Thank you.
Mm-hmm.
Sishu in the chat also asks, I do have a question about the noise at the end. Isn't there correlated error that can arise to what you were talking about? Sorry if that's very vague. I don't follow all the details of the math.
Okay. Yeah, so noise can make even a stable system look like it's chaotic. A lot of times, I gave an example of the pacemaker circuit here. And come on. This is a live cell recording. Like, these are biological neurons. You can see that there's noise down here. Like, there's jitter. The ions are moving chaotically. Like, that's happening in the environment, but you still have predictable dynamics. So this is a question of controllability. You're thinking of the neuron itself as a controller, or of the neural circuit as a controller, or maybe some upstream neural circuit that's sending outputs to the neural circuit as a controller. And so some of these systems are controllable in the face of noise, some are not. Just depends. But yeah, if you have a chaotic neuron and it's quasi-stochastic, you perturb it, it loses chaos, then you can perturb it with noise, but the noise is stochastic. It's modeled by a stochastic process. And so it will induce chaos anyway, most of the time. There are examples, I can't think of any particular, like what neurons they are or what animal they're in, but there are examples of endogenously chaotic neurons that are stabilized by noise. A good example of this is the pendulum. If you face it upright and you oscillate it, you make it jitter up and down, it will gain stability in the aforementioned unstable equilibrium. So that kind of thing can happen with neurons too.
Great, thank you. Do you mind linking the blog post in the chat?
Yep. I can link to the slides. I'll do that after I go down here. Yep.
Thanks. Thank you.
Mm-hmm. Thank you.